Can you folks see the slides?
Yes.
Great, thanks. All right, so hello everyone. Thanks so much for joining us today. I'm just going to get rid of this control bar at the top. All right, so before we get into some project examples, I really wanted to share this quote from the Cochrane collaboration. It's about living evidence syntheses, and I think that it provides a nice introduction for what we're going to talk about today. So the quote reads, "The concept of living evidence synthesis and related outputs, such as living guidelines, are of increasing interest to evidence producers, decision makers, guideline developers, funders, and publishers as a way to seamlessly connect evidence and practice." So certainly during the past couple of years of the pandemic, we've seen research being produced at a rapid pace. And so the beauty of living evidence syntheses is that they allow you to capture a rapidly evolving body of evidence because they're continually updated with new evidence as it becomes available. So today, Leah and I are going to be presenting two different examples of living evidence synthesis products. And the first example is a series of evidence briefs, and the second example is a rapid review. So let's get into it. We'll start with the living evidence briefs. So to give a little bit of project background, the series of evidence briefs are on various therapeutic interventions for COVID-19. And we at the MSSU were first commissioned to do this work in November 2020 by the Nova Scotia COVID-19 Therapeutics and Prophylactics Advisory Group. That group is co-chaired by Dr. Lisa Barrett and Dr. Tasha Ramsey. And the group is tasked with providing recommendations to the health system regarding clinical use of COVID-19 treatments. So the evidence briefs that we at the MSSU develop inform the group's recommendations. And the overarching purpose of the briefs is to summarize evidence specifically about regulatory approval, clinical guidance, any safety advisories, and relevant news articles pertaining to the matter of interest to the group. And for these sort of categories of evidence, the group is interested in developments from within Canada, of course, but also in other jurisdictions that are similar. So the US, the UK, Australia. I'm just hearing a bit of feedback, but I'll just keep going. The group is also interested in study findings or status updates from key clinical trials, as well as any high quality evidence syntheses, such as systematic or rapid reviews. So the content and structure of the briefs are tailored to what the group needs to make their recommendations. So in that way, it's a very collaborative effort. And a bit more background, the briefs aren't systematic or exhaustive, but rather they're designed to be a snapshot of the evidence available at the time. And there are 15 different therapeutic interventions that are of interest to the advisory group, and each intervention has one brief, and they're updated roughly every 3 to 4 weeks on a rotating schedule. Because the briefs aren't systematic, they can be updated by one person. And again, this level of rigor and the process that we've etched out for continually updating these meets the needs of the advisory group. Finally, deliverables are shared with the advisory group, and they're also posted publicly to the MSSU website. So before I give an overview of the searching and updating process, I thought I'd talk just a couple minutes about some of the theory that informs the structure and the content of the briefs. So given the impact of the briefs on the advisory group's decision making, it's of course critical to choose reputable sources of high quality information. And even though we're not conducting an exhaustive search of the literature every time we go to update these, the focus is on searching a select number of key databases, as well as searching for Grey Lit from key and reputable authoritative bodies. Just touching on preprints, over the past couple of years, we've seen many issues with preprints, such as the fact that they're not peer reviewed, they can be shared widely and misinterpreted and misrepresented in the media, and also how they contribute to misinformation. For that reason, And in an effort to cut out the noise and only synthesize the best evidence, we do include preprints in the briefs primarily when they report on key clinical trials. And finally, our approach for searching systematically in Google is informed by the article titled "Applying Systematic Review Search Methods to the Grey Literature." So this is an overview of the searching and updating process. Each brief takes approximately 1 to 2 hours, and the focus is on any news or developments since the last update. So I always begin by searching in the databases Medline and Epistamonikos. And in Medline, I use a targeted search, and then the searching in Epistamonikos is manual. And so the goal is to find articles that are published in quality journals or preprints that report the findings from key clinical trials. Next, to find that gray literature, I use Google using various combinations of search terms plus advanced searching commands. And typically the first 3 pages of results for every search are screened. And the sum of the categories of GreyLit that the group is interested in includes press releases from drug companies, safety advisories from public health agencies, any sort of clinical guidance or reports from authoritative bodies. So that could be CADIF, Alberta Health Services, etc. And we're also interested in news articles that speak to the approval or use of a drug or that report on preliminary findings from key clinical trials. A bit of feedback.
No different than anybody else. January stuff is after Christmas.
I'll carry on. The next, because the briefs again aren't systematic or rapid reviews, there's no formal screening or extraction, but rather screening and extraction is done informally and simultaneously as I search through the evidence. And then the final step in the process is compiling the deliverables. So that would be each individual brief. And that entails incorporating any new evidence since the last update into the brief. And as we'll see in a moment, any new updates are highlighted at the top of the brief. And then for included studies, the main characteristics, results, and conclusions are presented as bullet points, and then other forms of evidence are mostly narratively summarized. This is an example brief. This is the one for ivermectin. And as you can see, this particular version was last updated on October 25th. So since then, there may have been more developments. But as you can see at the top of the brief, there's an overview of the drug. Next, there's a new in this update section where newly added evidence is highlighted right up front. Typically, the next section is about drug approval, where information about whether or not the drug has been approved in any particular jurisdiction is included. And again, we're interested in Canada and any other similar jurisdictions like the US and the UK. If the drug has been approved, then we typically detail in what population it's approved for, in what dosage, and any other sort of relevant clinical guidance. Also, if there have been any safety advisories issued for the drug, warning against the use of the intervention in certain populations or what have you, that information would also be included in the approval section. And then next, I have just some more screenshots of various sections of the brief. And for the most part, the briefs are structured the same. In this case, with this example being ivermectin, we know that that has been a really hot topic in the media. So in this particular brief, there is an actual media section in the top left-hand corner. You can see it there. And that just draws attention to some news articles about the misuse of the drug. Another section of this particular brief is the evidence synthesis section. So in this case, we have some high quality, either systematic or rapid reviews that are highlighted, including their conclusions. And so there's a Cochrane review in this case, an evidence report by Alberta Health Services, and then a health technology review from CADIF. that sort of gives an idea of the level of evidence that we're including in these briefs. Again, only synthesizing the best available evidence for the particular drug. In the top right-hand corner, you can see there's also a section on clinical trials. And again, this section highlights the main study characteristics and findings / conclusions for key clinical trials. And just in the bottom right-hand corner, I wanted to provide a snapshot just of the last page of the brief. You can see that the references are there, but I also want to highlight the fact that we have a small, albeit method section where we detail the specific date that we searched and the sources. And this is really just to show that even though, again, we're not conducting a systematic or rapid review every time we go to update these, it is really important to be transparent about the methods that you use. Regardless, and especially when the product is going to inform decision making in any way. So that's one example of the briefs that we do, 1 of 15. And so next I will pass it over to Leah.
Awesome, thanks, Christy. So now I'm going to move on to a living rapid review that we've been working on for the better part of a year now. And this is a more formalized review. So this is more similar to a systematic or scoping review, except very abbreviated, as I will talk about. So next slide, please, Christy. To give a bit of an overview of the project. So the topic of this rapid review is the public health implications of SARS-CoV-2 variants of concern. When we say variants of concern, we mean the ones that have been identified by the World Health Organization. So anytime you hear about Alpha or Delta, we're talking about these VOCs or variants of concern. And this project was first commissioned back in February 2021 via COVIDEnd, which is it's the Canadian national branch of a global network that is intended to coordinate evidence synthesis response to COVID-19 around the world and across the country. And they identify rapid review teams across the country to work on topics that have been identified by key knowledge users. And they use the Spore Evidence Alliance's established network to tap into those teams. So both COVIDEnd and Spore Evidence Alliance were sort of brokers for this request. And the initial knowledge users were Alberta Health Services and the Public Health Agency of Canada, sort of as secondary knowledge users at that time. Then in the summer, we received additional funding through COVARNet, which is a newly established, again, sort of evidence synthesis network. This time it's focused on VOC specifically. So they were able to extend our funding in July 2021 through to the end of this year. So we've been working on it with a plan to reassess if it will be continuing in December. And now the Public Health Agency of Canada is the main knowledge user. So they are the ones that we are liaising with during this process to respond directly to their needs related to this topic. And so the purpose of the rapid review, the living rapid review, is to summarize evidence about 3 types of public health response to variants of concern. The first is modifying approach to vaccination. So in this case, we're not talking about vaccine effectiveness, attitudes towards vaccines or vaccine uptake. There are other teams that work on those topics. We're focusing more on the approach to vaccination, so things like timing of doses, mixing of types of doses, third doses and boosters, administering to children, that sort of thing. Next, we look at infection prevention measures, so things like masking and physical distancing as they relate to variants of concern. And then we look at infection control measures, so things like quarantine, lockdown, testing strategies. Of particular interest lately to the Public Health Agency of Canada have, of course, been children in schools. So we've been trying to really make sure that we're capturing all of the evidence that specifically pertain to those kinds of settings. But really, we're looking at any kind of community setting in which these types of public health response are occurring. Next slide, please, Christy. So for the schedule, this rapid review is updated every 6 weeks, but it's actually done on a 3-week time frame. We alternate it with a jurisdictional scan component, which unfortunately I won't quite have time to get into today. What that jurisdictional scan does is we look at public health guidance that has been issued by provincial and territorial governments and some other national governments from around the world as they relate to variants of concern. So we do that deliverable as well as this rapid review, alternating, each done within a 3-week time frame. So we're doing this rapid review in a 3-week time frame. And the deliverables are posted to both COVIDEND and Spore Evidence Alliance's website. Later on in the slides, there will be links to those, and these slides will be made available after the presentation. So any links that are in here, you'll have the opportunity to check out afterwards. And for the rapid review, our deliverables include a full report, and then a summary table of evidence, which is a supplementary file that includes more details about the studies in case the reader is interested to go in and read more about what the studies are. So we provide that as a supplementary file. And we also provide a one-pager. So of course, the people that we work with at the Public Health Agency of Canada are very busy. And so we try to distill the information as best we can into a one-pager so that someone who is very busy can take a look and see what the latest evidence is about this topic. Next slide, please. So I'm also going to talk a little bit about the theory to start behind this. So essentially what we are doing for this Living Rapid Review is a modified Joanna Briggs Institute, or JBI, approach to systematic and scoping reviews. We're doing a little bit of a hybrid of a systematic and a scoping review, which I'll get into a little bit more. We do include quality appraisal, so it's not quite a true scoping review, but we don't do any type of formal synthesizing or meta-analysis of the data. We are just summarizing what we find. So it's kind of a little bit of a hybrid. The Spore Evidence Alliance, when they commission teams to do these projects, they actually offer a training session on rapid reviews to every team that is working on them. So that was a great session to attend. And they're also offered to patient partners who are involved in the teams. So we do have 2 patient partners who are on our team. And I'm going to talk a little bit more about that relationship in a moment. And that training is based on a guide by Trico, Langlois, and Strauss that is through the World Health Organization. It's called Rapid Reviews to Strengthen Health Policy and Systems, a Practical Guide. If you haven't encountered this before, it is a really, really great resource. It's sort of my touchstone resource for working on rapid reviews. It talks about how best to develop rapid reviews depending on your audience, particularly when it comes to health policy and systems, different shortcuts you can take, how you maintain rigor, all that kind of stuff. It's all in this really, really great resource. And our approach is also informed by a paper that I was a co-author on that kind of puts this on the backdrop of COVID-19, different challenges that we have faced during COVID-19 as people working in evidence synthesis, especially working on rapid reviews. So that's another paper that you might want to take a look at if you're curious about rapid review methods, particularly during COVID-19. Next slide, please. So key strategies for this rapid review in particular, the first thing was to plan ahead. So we actually developed a work plan that we submitted to both the brokers and our knowledge users in advance of starting to lay out our plan. And that included which shortcuts in the standard sort of systematic or scoping review process that we were going to take and how to take them effectively and transparently. Transparency is so important when you're taking shortcuts in rapid reviews. It's really important to let readers know if you've been cutting corners, which is totally admissible during a rapid review because the timelines are so short. It is very, very accepted that shortcuts have to be taken in the standard systematic review process, but it's just really important to be transparent about what those shortcuts are. Then we assembled the team, and I can't stress enough that the team for a rapid review with this short of a timeline really, really has to include at least one member that has extensive knowledge and experience in evidence synthesis methods. So this is not the time for a project co-lead to be learning on the job. The timeline is just far too short for that. We certainly do have many trainees on the team though. So we have a core team. So I'm co-leading this project with Dr. Janet Curran. And then we have 3 other team members. We have a project manager who works at the JBI Center at Dow. We have a former project manager who's still on our team. We have another postdoc, and we're sort of what we call the core team, who sort of lead the team, meet weekly, make sure that we're liaising with FAC and other knowledge users as needed with our patient partners. And then we have a kind of rotating group of, I'd say, 5 or 6 screeners and data extractors who help us out as they're available as we go. They track their hours, and some of them are faculty who have time on their plate and have been wonderful to help. And some are trainees, master students, doctoral students, postdocs. We have an evidence synthesis fellow from Ireland as well, who is on our team, who is learning. So it's been a wonderful learning opportunity for them. So this isn't to say that that trainees can't participate in rapid reviews. In fact, it's kind of like trial by fire. It could be really, really, really good. But the core team really, really has to have experience in knowledge and evidence synthesis to keep this bus rolling on time. Having such an interdisciplinary team has really been an asset, I think, for the project. So I would say really none of our core team members even have significant expertise in public health. We come from all different areas of health. I'm a librarian, of course, but that has sort of allowed us to have a bit of a subjective view of the topic area, and we're able to present the evidence as we see it. And so that's kind of been an asset for our team. Now, I know we have a lot of librarians in the audience today, so I will give us a shout out that having an evidence synthesis librarian is really, really also key to this process. Having someone who knows what they're doing with getting to the databases, with exporting, with tracking, making sure the reporting is all in order, really, really important to this rapid review. And of course, our patient partners, so they add a different perspective. COVID-19 affects literally all of us, and it's really, really important to have patients on the team lending their perspective. Now, I will say it hasn't been easy to have patients engaged in this rapid review process. They weren't able to help us decide on the topic matter because they were given to us by our knowledge users, by Alberta Health, and then by FAC. So they weren't able to really add their perspective to that. What we've managed to do at this point now is that once we finish our deliverables, we give them the deliverables before we deliver them, and they provide patient-identified key messages for us to share with our knowledge users. And that has, you know, when you're working so deeply on a project, it can be nice to have someone from the outside say, okay, this is what I'm seeing in the big picture. This is what's really important to me. And they also attend our weekly meetings as they're able as well. So they are involved in sort of the behind the scenes planning, even though this is such a rapid process. So it's been really, really important to foster these relationships with a whole bunch of different groups. Our knowledge users, the knowledge brokers who commission the projects, our other research partners. So as I mentioned, the JBI Centre of Excellence at Dow, Evidence Synthesis Ireland has been the one who has facilitated this fellow who has been helping us on the project. And of course, those patient partners. So it really is about relationship building and making sure that the product is appropriate to the needs of everyone that's involved. Next slide, please. So this is essentially the life cycle of the review, and this is based on, loosely based on an infographic by Evidence Synthesis Partners. And it truly is a cycle because we are updating it every 6 weeks. And it starts with not defining the research questions, but redefining them. So the initial research questions were identified quite a long time ago, but we are constantly tweaking different ways to approach. We meet with FAC every 3 weeks after the latest deliverable goes in, and we say, is there anything else that you need us to pull out? Is there any different way that we could organize the information? Anything like that. Then I run the search on day 1 or 2, depending on if I have to change it. I haven't really changed it since Delta emerged. That's the last time I really changed the search. So I run that on day 1 or 2 and get that all into Covidence, and then screening begins as soon as possible. We work on both title and abstract and full text screening the first week. The second week is dedicated to data extraction and quality assessment. And then the third week is dedicated to compiling those deliverables. Next slide, please. So to get into each one of those steps a little bit more detail. So as I said, when we're talking about defining or redefining the research questions, that work plan was designed before the project started. We do have regular meetings with FAC every 3 weeks to talk through what they're looking for in this round. And we make tweaks to the report tables almost every update. We're changing something or some way that we're extracting data or presenting data. The search is run in 5 places. We run it in Medline, Embase, the Cochrane Library, which includes the Cochrane Database of Systematic Reviews, and Central, which is the Central Register of Controlled Trials. Epistemonikos, as Christy mentioned before, they have a living overview of the evidence related to COVID-19, so we look in that. And we look for preprints in medRxiv, which has its own little quirks, which I'll talk about in a second. We use Covidence for our screening process, and we do use Covidence's deduplication function. I'm well aware that there are many other ways to do deduplication, especially using EndNote and things that are kind of have more control over that process. We don't really have the time or resources to do that within our 3-week schedule, so we rely on Covidence's automatic deduping function. We had experimented with doing graylit searching early on in our process. We found that it was a lot of effort with not a lot of return, so we weren't really including many resources from a graylit search of Google or specific websites. So for the purposes of keeping this process really streamlined, this is one of the shortcuts that we have taken, is that we are not looking for graylit specifically other than the preprints that we find in medRxiv. So the screening is done in Covidence. It is done in duplicate. So we have 2 separate screeners doing each phase of screening, and then we resolve any conflicts. We tend to use a pretty broad inclusion criteria for the title and abstract round. So the search is designed to capture all literature related to variants of concern. It doesn't have any further hedges related to public health measures. We just want to capture everything. In the beginning, which I'll show you, in the beginning, this was easier. There wasn't as much literature about the variants of concern. That has changed, so the approach may have to change. But for now, the searches are just designed to find everything related to VOC. And another thing I want to highlight is I do not do bulk full text retrieval. I do not pull PDFs and attach them in Covidence. We don't have time for that. One of the things about COVID-19 that's been really lucky is that much of the material is actually open access that's related to the pandemic. So we are really easily able to click DOI links in Covidence to access full text. There's really no need to be pulling PDFs in mass like that. It just takes way too much time. Our data extraction and quality assessment. So our data extraction is done by a single extractor, and then it is verified by one of those core team members, so one of the 5 of us. So we don't do double data extraction in duplicate independently and then verify it, because that would, again, just take way too much time. This is by far the most time-consuming aspect of this work, is the data extraction and the quality assessment. Initially, like early in this project, most of the studies we were finding were modeling studies, because there just wasn't that much real-world data yet. That has changed. We're finding a lot more real world data is out there, more observational studies. So we're doing more and more quality appraisal. And we use JBI quality appraisal tools for that. And then when we compile the deliverables, we put those key messages up front, and we prioritize a table format for everything else. So we're not looking to provide big chunks of text. We are trying to distill a lot of information in as easy to read format as possible. And we compile those tables while we're doing the data verification. So one of our extractors extracts the data into Excel, and then as we're verifying, we go through, make sure that that data extraction is accurate, and then right away we're plugging it in, building in those tables for the report at the same time to save some time. And as I said, we put more details in the summary table that people can look at if they want to. Next slide, please, Christy. So again, I know there's a lot of librarians in the audience, so I wanted to touch on searching just a little bit in a little bit more detail, just in case you're curious about that kind of process. So when I run the searches in databases, I run them from the beginning of time for every update. So I am not running from the date of the last update. I'm running from the beginning of time. This is because you really don't know when things are added to databases and how they're dated. So there may be things that are backdated, retroactively added, retroactively indexed, things change. So I want to make sure that I'm capturing everything every time. And then I use Covidence to remove the duplicates. So first, I remove internal duplicates within the set. I have a separate Covidence project for this that's not connected to the main project. it's my deduping library is what I call it. And I use it to dedupe that set for that update. And I use Excel to track the number of records retrieved, of duplicates removed. I don't rely on Covidence for this because we're adding to it all the time. The numbers become inaccurate very quickly. So I track that myself in Excel. We don't include a Prisma diagram or anything like that in these reports, but we have written a couple of manuscripts. We do plan on future publications. So it's just really important to keep track of this in case we ever do need to report it in the future. And then after that set is independently deduped, then I add it to the main review in Covidence. And that's where the overlap, the overlap with the previous searches is removed in there. So we're not screening anything twice. Covidence automatically removes anything that we've already looked at, but now we can be sure that we're finding everything. Now, medRxiv is a little bit different. medRxiv doesn't handle systematic searches very well, as I'm sure anyone who's tried to use it during the pandemic has learned. It doesn't support multi-line searches, and I haven't really been able to get it to export any more than like 50 records at a time, which can be very frustrating. So it's kind of troubleshooting with medRxiv. So in this case, I do only run updates from the date of the last date. So it's a little bit different from the other databases, but that's how I incorporate medRxiv results into the search. Next slide, please. And I just wanted to share some numbers with you. So is this sustainable moving forward? So as I said, our funding is up in December, and we're not sure if it will be extended. It might be extended. Chances are this topic is going to remain relevant for the foreseeable future. But I just wanted to show you the cumulative number of records that we have retrieved over time. So we started with just over 1,000 records back in February that were related to variants of concern. And today, I ran the last search this Monday. We're at over 5,000. This is in all of the databases combined. We started by only including 18 studies, and we're now up to 126. And I will say that anywhere up to 80% of these are preprints. So Christy mentioned issues with preprints before. We have no choice but to include preprints in these reviews because they form such a large part of the literature. But we do, that's part of why we do a quality assessment as well. And we do sort of dock a point from our score from preprints. And of course, we also do other aspects of quality assessment too. So we do kind of try to mitigate the fact that there are so many preprints included, but we have no choice but to include them. So who knows how many will be included in this latest update. We'll see if it continues to be a steep curve, but yeah, I don't know what the future holds for this project, but we'll find out. Next slide, please. I wanted to provide some snapshots, and there's a link. Again, these slides will be made available. You can click through to see the full thing. But this is just a snapshot of one of the evidence tables that we include in the report. So every time we add new information in an update, we put it in bolded blue font so that it's easy to see what's new since the last time we provided an update. We have it divided by VOC. And then if there is information that is relevant to more than one VOC, we just we repeat it across multiple columns. And as I said, short, snappy points are preferred. So we try to distill complex information without stripping too much detail from it, but making it as easy to read as possible. Next slide, please. This is a snapshot of the one pager that I was talking about. So these emerging points of interest in this blue box here, they're the same ones that we provide at the top of the main report as well. So these are sort of the main things that we want to communicate to our knowledge users. We put them here. And then along on the right-hand side, we have abbreviated objective, background, and method sections. Just in case this is the only thing that they ever see, at least we have some information of how we got the evidence that we're presenting. Next slide, please. And finally, I wanted to show a snapshot of some of those patient identified key messages that I was talking about. So they go directly under the emerging points of interest in our report in a separate box. So it's really, really clear that these are patient identified And they really have been really, really effective at communicating sort of the meat of what we want to share with our knowledge users. It's a way for patients to have a direct line to the Public Health Agency of Canada, and that's pretty awesome. So we're really, really happy to be able to include these patient identified key messages. All right, so we kind of went through quickly because we wanted to leave lots of time for questions at the end. So just some concluding thoughts. It's really, really important to choose methodology that is appropriate for the goals of whatever project it is that you're doing. So as we saw with Christy's examples of evidence briefs, not everything requires a full systematic review every time. But if you are running targeted searches, it's really important to aim for the highest quality sources as you can, so that you're presenting the highest quality evidence that you can. As well as sort of develop a protocol for yourself so that if you're running frequent updates, you have a system in place so that you're hitting the same places every single time you approach an update. You know where to look. You look in X, Y, Z, and then you know that you're going to find the evidence that you need from those places. I can't highlight enough how important transparency and methods reporting is. As I said, it's completely reasonable to take shortcuts in rapid reviews. That's how they're able to work. But it's really, really important to be transparent about what those shortcuts are, what you're doing. It has been really, really important to tailor the report format and language to your audience. So people at FAC, people in this advisory group that Christy is supporting, maybe don't have a lot of time. And so we've learned to be really concise with our language. Not everybody has time to read big blocks of text. And so making products that are tailor-made for your audience is really, really important. And streamlining and overlapping steps of the process as needed is also really important. So in the evidence briefs, Christy is searching and compiling sort of at the same time. So you're not doing them separately and taking more time than you need. And as I said, in the rapid review, we do parts of the data extraction and building the tables in the report also at the same time. Any way we can save time without compromising quality, we try to do it. And yes, it can be stressful, and there are some late nights and there are some stressful weekends, and that's the way it is sometimes, especially in this pandemic environment. But that's the way it is sometimes, and hopefully the work that we do leads to positive change in the health system. So now it's time for questions. You can contact us with your questions if you want follow-up. Our emails are there. I'm going on mat leave in, well, I don't know, hopefully a little less than a month. Hopefully I make it that long. So I'm available till then, but you can always reach out to Christy with questions as well. And these slides and a recording of the presentation are going to be on the website after this, and I believe in your emails as well. So yeah, thank you.
That's right. We'll send out the slides and also the recording after this, so you can, if you want to check it out, you still can. And we're going to have about 15 minutes for questions, and they're already coming in. That's great. And so what I'll ask you to do is to either type your chat question in the chat box and I will read it out, or if you prefer to verbally say your question, you can write your name in the chat box and then I'll read out your name and you can unmute yourself and say your question. So the first question is, who writes the summaries given to clinicians? Is it another member of the team or the librarian?
Yeah, sorry if I didn't make that clear. It is me as the librarian. So I'm searching, extracting, writing, compiling, and then that's all done by one person.
Thanks, Christy. So the next question that just came in is, how do you engage patient partners?
Yeah, it's a great question. So as I mentioned, ideally in an evidence synthesis project, and any project really, a patient partner would be engaged from the very beginning. So they would help you to identify your research question. They would help you to design the work plan or write the protocol, identify things that they would be interested in extracting and things like that. In this case, because it was so rapid and the questions were essentially handed to us by our knowledge users, we didn't really have time to do that. And so in this case, what they do is we provide them with what we have found and we ask them what they think about it, and they identify key messages as they see fit. And also they attend our weekly meetings as they're available. And that's how we've kind of managed to get patient engagement in there.
Thanks, Leah. The next question is, how do you know the quality of the data is good enough for clinicians to make medical decisions off of?
Yes, another great question. So I think my role in synthesizing the evidence is really to cut out the noise, cut out the things that we know are of poor quality or that haven't been proven to be of good quality, kind of package that up into a synthesized document. And then the advisory group, they're the clinicians who are going to be pulling in their experience along with other elements to make the recommendations. So, you know, the evidence briefs are a part of that. Again, it's like that small piece of evidence-based practice, but they are also bringing in their expertise as clinicians to decide whether or not, okay, well, the findings from this preprint, you know, do the conclusions measure up? Should they be implemented? Like, how does it measure up with other evidence? So I hope, does that answer your question?
That's great. Thanks, Christy. So there's another question here. Do you do a set of test screenings each time to verify that the inclusion criteria are still working for all team members?
So we don't have time to do a formal one every time. Earlier on in the process, we did a little bit more of that. But we do occasionally. So for example, we found that people were including more and more stuff that wasn't actually relevant to variants of concern. That's kind of the biggest way that things slip through the cracks. And so we have kind of a gathering moment through email, or we schedule a meeting to sort of chat through these issues and say, hey, we're noticing that a lot of non-VOC stuff is making it through. Just a reminder that we really want to make sure that it's related to VOCs before we include it. And so that's kind of how we do that. Even though we don't have time to do tests every time, we just kind of remind people of what the inclusion criteria is every once in a while.
Thanks, Leah. So there's another question about patient engagement. Do you change patient partners between reviews? And how do you choose your patient partners?
So for this, we've had 2 patient partners that have been with us the whole time. So we're not changing them up. They're kind of attached to this project. The Maritime Spore Support Unit has a network of patient partners. We have a patient partner group that we draw from. So we post opportunities through email to them, and they're able to volunteer. And then also Janet Curran, who is my co-lead on the project, she had a patient partner that she's worked with previously, and so she identified her. So it's kind of a little bit of personal networking, a little bit of networking through our organization that we're able to identify them. But for the most part, they self-identify. So it's always presented as a volunteer opportunity, and then they're able to volunteer if they're interested.
Okay, great. And so we have another question a bit about the structure of the project. Have other provinces used your evidence briefs and rapid reviews to avoid duplication of work?
That's a great question, and I'm actually not sure.
Yeah, I don't know about the evidence briefs. For the rapid reviews, part of the beauty of COVID-End as an organization is the point is that they are there to avoid duplication of work. And so the idea is that they're sort of overseeing this evidence and this activity across the country, and they select teams to answer questions so that hopefully there's only one team working on a question at a time. But yeah, the evidence briefs, we do post them online. So they should come up with, like if people were searching for things. We do acknowledge that there's probably lots of duplication of effort, but in this case, we're tailoring it to our particular audience and we know what their needs are. So that's how we kind of approach that.
So you just answered the next question, which is where do we find the evidence briefs? Are they available online? And maybe we could add a link to the email that goes out after this with a link to that. So if people want to check them out. No,
Yeah,
and there,
Rachel
Rachel's also just posted it in the chat for you.
Great.
Awesome. Thank you, Rachel.
So feel free to type your questions in the chat if you have them. I was just curious, actually, Christy, if you could talk a bit more about the media section of the brief that you discussed. I thought that was a really interesting thing to add to a brief that I hadn't seen before. And I was just wondering if you could talk about the decision to do that and how that's helped and been useful for you.
Yeah, that's a great question, Scott. So there's not a media section in all of the briefs. It really is ivermectin and maybe a couple others that have really been hot topics in the media. Certainly news articles aren't always going to capture what the group needs, like what they've identified as their priority sort of categories or buckets of evidence that they need to make their recommendations. So for the ivermectin example, the news articles that are linked in the brief really are those ones that sort of show how the drug is being misused or how misinformation is perpetuated about the drug's use. So there's some CDC articles. I think for the most part, the news articles are contained to stories within Canada.
Okay.
But for folks like the advisory group making decisions based on the briefs, sometimes knowing sort of public perceptions of how a drug is used or perceived is of interest. So again, there's not a media section in all of the briefs, but certainly for ivermectin, as we've seen, it's really important to sort of hit or to capture those stories.
And
Okay.
if I can add to that too, I didn't have time to get into this jurisdictional scan component today. As I mentioned, the one where we're pulling public health guidance from the different provinces and territories. What we have found is that more and more, especially around vaccine mandates, government websites are not updated in time. And the only place that you can find information about this are in the press briefings that they give to the media or in the follow-up CBC article that accompanies that press briefing. Sometimes you have to rely on the news media for some information, which is unfortunate, but sometimes that's all there is as proof of something.
Right. Yeah, I guess everything is moving so fast, it's hard to find the information you need. No more questions are popping up. I have one other question for you, actually, Leah. I was just curious about, you talked about the shortcuts and sort of deciding what those are. And I was just curious how you make those decisions and if they're sort of like decisions that are normally made and what you do.
Yeah, that guide that I was talking about rapid reviews, they have a really awesome table. And I'm not going to remember the page that it's on in there, but if you look at the report, you can see they have a table that kind of presents different options of ways that you can cut from the process and what the implications of those cuts are. So it's pretty common to search fewer databases than you normally would for a systematic review. And it's pretty common to have only one screener at either the title abstract or the full text stage and have one extractor. Hopefully, ideally not every single shortcut would be taken, but it's a really, really great table in that report that just kind of like talks through the options of how you might streamline that process a little bit. And for this one, we thought we'd take a lot more shortcuts than we ended up taking, but in the end, we haven't taken that many, in the grand scheme of things. We try to do as much as we possibly can within that 3-week time frame, which is a little bit madness, but yeah, we just we don't take a huge number of shortcuts.
Awesome, thank you. So there is a question here. It says, Christy, you mentioned poor evidence to eliminate. How do you know what evidence is poor? Do you have a criteria? Is it based on study type?
Well, certainly our focus is on key or core journals. So if a study is published in, JAMA or the New England Journal of Medicine, that's something that we look for, as opposed to study findings being reported in a very obscure journal. So again, with sort of the asterisk that the briefs really aren't exhaustive, but we're pulling together sort of what we consider to be the best available evidence, again, published in those high quality journals. And our focus isn't so much on study type, but pulling in preprints, again, when they report on the key clinical trials. So a snapshot of the evidence as opposed to everything. Does that answer your question?
And I would add to that it's more of an inclusive process than an exclusive process. So we're not finding everything and then excluding bad stuff. It's more like seeking out the good stuff to begin with, so that it's not really kicking stuff out. That's why we look for evidence syntheses too, because they've done a lot of that work people who have had the time to do actual full systematic reviews, they've done that work. So that's why we look for those too.
Thank you. If anyone else has another question, we probably have time for one more if you want to throw it in the chat. I'll just give you a minute to do that if you'd like. Okay. I'm not seeing anything else come in, so I'd just like to thank Leah and Christy for their incredible presentation. It was so timely and informative, and it was so great. We're getting some positive comments in the chat about that as well. And I'd like to thank everyone who attended today and contributed to our discussion. And I'd encourage you to attend our next one, which will be in 2 months. I'll also be sending out an email, as I said previously. It will have an evaluation survey that we hope you fill out. Let us know what you liked, what we can improve. If you have any topics you're interested in hearing about, that's really awesome to hear. And we'll also be sending the slides as well as a video of the presentation. So thank you so much for your time and have a great day.
Thank you, everyone.
Thanks, everyone.